Well, thank you very much, Alison and James, for this organization, first of all, and also always lively discussions going on over here. So I really enjoyed the last talk so far, and I hope I can live up to the expectations. Hope I can live up to the level. So I'm going to talk about how we can regulate molecular assemblies, clusters of molecules coming together, using chemical reaction cycles. So not chemical reactions per se, but really a cyclical reaction. And let me start with that first, why I think that is important, that this is cyclical. And this comes from biology. So why would we want to use a cyclical reaction rather than just a reaction, A becomes B? And the simple answer is maybe the unsatisfying answer is because biology does it too. So I give an example over here, which is the ATPase. Calcium 2 plus ATPase. And as its name suggests, it hydrolyzes ATP and it converts one molecule of ATP by reacting it with water into ATP and inorganic phosphates. And it doesn't do that for free. It uses the energy that it gains upon performing this reaction by pumping across some molecules or some ions across a membrane. So two calcium 2 plus ions are being pumped from one side to the other side of the membrane. And it does this against a gradient. So there's more calcium on this side than on that side. So obviously that costs you energy. So you need to invest a little bit of energy. But what I want to focus in is the beauty of the cascade that happens and why this happens in a reaction cycle. First of all, we bind these two ions with high affinity, and then we phosphorylate, I think it's a glutamic acid derivative over here, by a reaction of this glutamic acid with ATP. So we are transferring, this is the first step in the reaction cycle, we're transferring one phosphate group over there. And that transfer comes from ATP. This is now going to a lower energy ADP states. And this changes the molecular conformation of the entire enzyme, the entire complex. This, of course, is a very complicated molecule, but it changes this conformation, and this allows the shuttling of the two calcium into that direction. Now, I don't even understand the full details, and I don't want to go into the full details of how that happens, because I want to focus on why we're doing this with the reaction cycle. But I think this is extremely elegant chemistry, and I wish we could build something like that in a lab. We're very far away from that. But then upon release of the two calcium ions on the other side, it also gets dephosphorylated. So it removes the phosphate. And the important part over here is now we're back at the starting position. So we can now undergo this cycle again. So this is a chemical reaction that proceeds in two steps. First, ATP, transfer of the phosphate, and then release of the phosphate to go back to the original state. If this was not a cyclical reaction, so this is just one reaction and it transfers it, you would have to resynthesize a new enzyme every time you do that. And that would be insane, of course. So I want to stress over here that the enzyme is just a catalyst that transfers ATP into ADP and performs a function while doing the entire reaction. So that gave us inspiration of, okay, can we now build something similar? If we want to go to molecular machines, then we need to build similar principles. And I know that I cannot build such a complex machine yet, but I know that we have to look at this fundamental principle, the use of reaction cycles to change functions of molecules. So in very schematic chemistry point of view, what we're looking for is a catalytic reaction cycle where a chemical fuel, I call that, so ATP, a high energy molecule, activates a molecule to do a certain function, but it doesn't remain there. That will be just a chemical reaction. It dephosphorylates or it reverts back to its original precursor state. And by doing this, it allows you to use that molecule hundreds, thousands, or hundreds of thousands of times. Every time one molecule goes in, it converts chemical fuel into it of waste and goes back again, performing its function over and over and over again. So that's what we're looking for. How can we build something like that as simple as possible? And that's what me as a chemist, as a supramolecular chemist, is interested in. So we built several reaction cycles. This is some older work that I did in my PhD, where we used carboxylates, and carboxylates were our precursor. And our chemical fuel was dimethyl sulfate. Dimethyl sulfate can react with this carboxylate to form its methyl ester, but this spontaneously hydrolyzes back again. So we now have a catalyst over here that converts this chemical fuel into waste over there by reacting, and the second step, waste over here, and undergoes one reaction cycle in which we effectively convert one molecule of dimethyl sulfate into monomethyl sulfate, by reacting it with OH minus, which is hydroxy, releasing another methanol molecule. So in one reaction cycle, we've temporarily changed this molecule. We methylated it rather than phosphorylated it. We methylated it, and it can go back again. So this molecule can undergo hundreds of cycles, thousands of cycles without any side reactions. It's a catalyst. It converts this fuel by reacting it with that into this state and that state. And then we couple that to self-assembly, so self-assembly into fibers. This reaction cycle was slow, in the sense that this molecule has a half-life of several hours and kind of nasty chemistry, and it has to happen at pH 11. So when I started my own group, I'm still interested in this idea of can we make reaction cycles and can we now make molecular machines with this kind of reaction cycles? First, looking at can we develop more reaction cycles that are a bit more versatile and a bit easier to handle, so that also the rest of the community can work on this idea. And we came up with this reaction cycle. I like it a lot because it's extremely versatile, and we're using it for many problems or many challenges, how you convert chemical energy into something useful. And it works with a dicarboxylate, and the dicarboxylate in a two-step process can be condensed into its anhydride. And condensation in this case, in the field of chemistry, means we're removing a water molecule with a condensing agent, carbodiamide. So I call it a chemical fuel. That is our ATP analog, a high energy molecule that gets converted into waste, urea, in a two-step process, and gives us an anhydride. An anhydride in this context means that without water. So we remove the molecule of water, and we're doing this in water. So the water is being reacted back on via a deactivation or hydrolysis reaction. So effectively what we're doing is we're hydrolyzing this molecule by reacting it with water to give us urea and temporarily activating this product molecule over here. Very similar to this idea of we temporarily phosphorylate something, we temporarily remove water, and then we put it back on. We remove water, put it back on. Very, very limited side reactions. So this molecule can go and undergo hundreds of thousands of cycles before side reactions start kicking in and before you get problems. It's a very versatile reaction cycle. It works in water, pH 5 to 7, pH wide temperature range. And this is aspartic acid. So it's a peptide. So you can use this on many types of peptides, of course. So you have a very wide range in your precursor space or your chemical space in terms of precursor and in terms of fuel. The anhydride half-life, depending on what type of anhydride you choose, you can tune that from tens of seconds to hundreds of minutes even. So that gives you also different speeds of this chemical reaction. Let me show you what I mean with that. If the chemical reaction cycle that I showed is our engine, this would be our gearbox. So we have molecules that are activated and only survive. This is residence time. So I have a residence time of 67 seconds, which corresponds to roughly 35 seconds half-life. And we have molecules that have a residence time in the range of 233 minutes. So it sits there for several hours. So that means the cycle is the same. It just has weak or unstable, and more stable active states. And that allows us to tune how fast the reaction is going. And it allows you to tune the activity of the behavior. So I like this particularly when you find an active behavior. Okay, this is driven by the reaction cycle. You can now tune the rate of this active behavior too. And you can immediately see, is it really an active behavior? Because if it's 10 times faster, if you're doing it with this reaction cycle, you know that it's really driven by the chemical reactions. So this is why we use chemical reaction cycles. And let me now show what we typically do to couple that to something useful or to couple that to a function. So we've been playing around with using then the R group in this molecule over here by making oil droplets, for example. And the oil droplets, we find self-selection principles. So molecules that are phase separating in the oil droplets, they have different half-lives than the molecules over there. I really come very shortly back to that later on. We find fiber molecules that we form molecules that form fibers, and these fibers, they show all kinds of dynamic behavior, motion, stuff like that. We find a principle that is called accelerated Ostwald ripening. So Ostwald ripening is the principle of droplets getting larger and larger with the function of time because they're thermodynamically more favored to be one droplet. We can accelerate it if you put a chemical reaction cycle on that. And then finally, and this is what I'll talk about in more detail, we couple that to the formation of compartments. I'll come back to that in detail. So how do we go about it? We got our reaction cycle, and then we start thinking, okay, we want a we want to do something with this reaction cycle. And in this case, we were particularly interested in chemically fueled emulsions. So these are droplets that are emerging because of the active state. So the active state can phase separate, but only the active state. So the emulsion will only be there when you add fuel, and it will only survive when you keep on adding fuel. Why are we interested in this? I think they serve as a model for membranous organelles. Membranous organelles are also droplets that we have in our cells. They are organelles without a membrane, so they're based on complex coagulation to form. And more research is pointing towards that they are also active state, regulated by phosphorylating dephosphorylation events. So it could serve as a model for that. It could serve as a model for protocells. They have a lot of lifelike behavior. They emerge spontaneously. They will die without fuel, and this shows self-division behavior. And then I think it's a key step towards the synthesis of life. And that's something that we're interested in. So we need a compartment if we would like to synthesize life, and we need a compartment to exist out of equilibrium. And that's exactly what we have in this case. So let me give you some details. So what we typically do as supramolecular chemists, we think of, okay, this is an interesting problem. How can we now design a molecule around that would answer, or that would work out the problem that we're thinking about? So in this case, we were wondering, could we develop molecules that are responding to our chemical reaction cycle by forming complex coactivate-based droplets? And complex coactivate-based droplets are droplets that are forming spontaneously through phase separation because two oppositely charged polymers or polycations come together. So in this case, we have a molecule that is 3+, and that is gluing together RNA, which is poly negatively charged. And it works like this. We build a precursor that is 1, 2, 3+, and 2-, and these are the two carboxylates that are in our reaction cycle. So this is the chemical motor, let's say. So a 3+, 2-, making it a +1 molecule. Switch ionic +1. And it interacts very weakly with RNA. It doesn't want to glue together RNA. But if we add now this condensing agent, we get a 3+ molecule, highly cationic, and it glues together the RNA very well. So this now sticks to the RNA, starts forming a complex coactivate through ionic interactions. That forms a droplet. However, that's a non-equilibrium state, and it only has 30 seconds. So in the 30 seconds half-life, it can be part of the droplets, but then it will get deactivated. It doesn't like the RNA anymore. It will leave the droplet again. So this droplet is now regulated through inflow of material and outflow of material, rather than sitting there in close to thermodynamic equilibrium. So all of these droplet properties now depend on the chemical reactions that are underlying this behavior. So what does that look like? First of all, we look at, okay, what does a sample look like? We make a solution, we apply fuel, and you see immediately it gets turbid. And over the course of 18 minutes, the system has burned all of its fuel. That's why that fast half-life is so nice. It has burned all of its fuel, and the emulsion goes back to its starting point. So you can now add another batch of fuel, and the emulsion emerges again. It's nice because the product plotted right over here nicely correlates, the concentration of product nicely correlates with the turbidity of that sample. So that also means that there's a very strong correlation between the molecules that are forming the droplets and the turbidity, the evidence of the droplets. So what do we see? We see that the emulsion emerges in response to fuel, and it decays without fuel. And then we grab a microscope and we have a look at what's going on. We did, in this case, specific case, we did it with microfluidics, which allows us to put all the ingredients in one bubble. Always a couple of 100 micrometers across, maybe 200. And we have that droplet ready. The moment it's produced, we capture it and we look at it, and it's roughly takes us a couple of seconds to make it, and then have in focus everything ready. And then we let it roll, and we just let this is a time lapse over roughly 20 minutes, where we saw the emergence of droplet. We're looking at the RNA molecule. These are the fluorescent molecules, but the peptide is in there gluing the RNA together. And we see all these droplets emerge, and we see them competing with each other. And as the system is now slowly running out of fuel, we see that the smaller droplets have died, and eventually the big fat one also dies out. You could now add a second batch of fuel, and then the whole system starts over again. So we see this emergence and this death, and I think in terms of synthesis of life, I find it's extremely appealing that you have something that spontaneously emerges, compartments, it's different than its neighbors, and it will die out if you don't take care of it. You have to keep on fueling it, just like your bacteria. We see all kinds of cool behavior that you would also expect close to equilibrium, like fusion, but also behavior that is really weird and something that's really out of equilibrium. Have a look at this droplet over here. I hope you see my cursor, the one that just emerged. And what you see, it started out as a bowling ball. It was completely homogeneously filled with RNA. and it converts into something that's more like a soccer ball, where the inside is actually composed of water. So you have water inside, water outside, but you still have the shell. What's happening over here is what we're suspecting is happening over here is that we still have fuel in the system. So the outside is still bombarded with new molecules that can hold together this RNA, but they don't make it to the inside anymore because there's not so much stuff coming in. So before they make it to the inside, or the inside molecules are already deactivating, hydrolyzing, and this is a pathway towards the death of this droplet, where basically the cavity, the inside of such a bowling ball or such a droplet gets dissolved first, and you end up with a shell that survives until all the last fuel is gone too. But not before, this truly hollow inside and you truly have water inside. They won't die before one last spectacle. As if we have a look at this droplet over here, you see that it starts to form this vacuole. And then, as I would expect it, that it would kind of like disintegrate and molecule by molecule leaves, but that's not the case. They die out by spitting out all kinds of I call them daughter droplets because I'm thinking of synthesis of life, of course, and it's not daughter droplets, it's just fragments of droplets. But these fragments of droplet, they survive for some time, for tens of seconds, maybe a minute, before everything dissolves eventually. So it's kind of like a very bad asymmetric self-division event. And when I think of self-division, something I really want in my droplets, you think of, okay, one droplet perfectly splitting in two. That's not the case. It just spits out all of these daughter droplets. But I'm super excited about this because this 19-minute mark is apparently exactly when we should add in a second batch of fuel. And can we save those daughters? And why did those daughters survive? Are they chemically different than their competition that did dissolve at this point? So these are the questions that we are now trying to answer. Like, is this just a homogeneous mess, or do all of the droplets have an individual identity? I think microfluidics is one way to go. So if we now look at such a microfluidic experiment, we can basically look at one of the later frames. See droplet A, B, and C, very original naming, but these are the three last droplets that survive. We can look at where did they come from? Like, okay, where did you originate from? Maybe we look from droplet A, for example. You see that it is coming from those two droplets. So it was one of the last fusion events until that droplet came. And those droplets, they came from all these droplets over here. So you can really see which droplet fused with what droplet at which time points, and what led to the death of certain droplets and what led to the survival of others. If you look at population D and E, for example, they don't make it to the end. It's not because they are absorbed by something else. They just died before their competition. Why did they die? Are they chemically different? Is there a different environment, or it's all the same? And of course, we want to steer that a little bit. What happens if we introduce a finite number of catalysts? Say put 5 particles in there that are catalytically active. Do the droplets that take up these catalysts survive longer than the competition or not? So can we offset that behavior a little bit? So what I want to show over here is that this active behaviors, individual droplet reach individual ages. So it seems to suggest that droplets have an identity. You can also see how far they travel. And as you can see, you have domain of orange, domain of green, and domain of blue. It seems to suggest that droplets don't travel very far for their food. They don't go all the way to the other side. Orange has eaten all this part. Blue has eaten all that part. They just eat their neighbors, but they won't cross all the way to the other end of the microfluidic droplets. Okay, so I want to change gears a little bit. Time over here. I started with this question for the motivation for chemical reaction cycles, and this is a molecular machine Foresight. So I also a little bit into molecular machines. We looked a lot at thousands of molecules coming together. But if you want to control, like biology controls over here, we have to start thinking of single molecules or much more defined molecular assemblies. So that's why we started to thinking about DNA nanotechnology. And even though this is not technology yet, it's very simple hairpin formation. We're working with, can we combine our reaction cycle with DNA, the precision of DNA? So we took some hairpins, and the hairpin is modified with a thymine base such a way that it can accommodate our chemical reaction cycle. And we took a molecular beacon that has a melting temperature in the range of 40 degrees. We start modifying this with a thymine base in such a way that we destabilize this molecular beacon. And indeed, we see that the melting temperature drops by 10 degrees if you introduce the thymine over here or over there, but not if you introduce it in the loop part. Then we don't see this drop. And you can add multiple thymines and it drops even further. So we've now destabilized our molecular beacon with this modified thymine. And now we're, of course, going to rescue it by the addition of chemical fuel. And the way that we express this is basically look at how many of our hairpins are folded. The folded fraction is roughly 50%. We're doing this at the melting temperature. And now we shoot in our chemical fuel and we see that we change the population of the folded hairpins. So that means that molecules get chemically activated. We know exactly how many molecules are chemically activated through HPLC experiments. And from that, we can derive that the fraction of molecule that is activated, 95% of them is in the folded state and 5% is in the unfolded state. So we're changing the position of how these molecules fold and unfold. And then we're adding a quenching agent that kills all the fuel by reacting with the fuel. It's basically competing with the fuel. And then we go back, not to the exact same state because there's a bit of buildup of waste. And this molecule itself is also not completely innocent. It also induces a bit of folding. It's very quick. It's very preliminary data, but we can now start building reaction coordinates and folding coordinates in an energy landscape, which is something that is way more complex to do with molecular self-assembly, where basically what we're doing is we've got a molecule that folds, unfolds with 100 millisecond time scale roughly. And it's roughly 50-50. So 50% is unfolded, 50% is folded. Each of these molecules get activated by reactive chemical fuel, shifting up the reaction coordinates. And there we have a different folding and unfolding states where it is 5 to 95%. So we now have a much clearer molecular picture. And even though this molecule just folds, it doesn't do any function yet. I think this is the pathway towards building molecular machines where this has a different function than the molecule over here. So we can really add chemical fuel. The molecule folds, does a function, opens up, and it's ready for the next molecule fuel, opens up, does a function, etc. So this is kind of my vision on how we can build this into nanomachines. I'd like to conclude over here. I have a couple of slides on this application. It depends on how we are on time. So what we did is we developed a simple chemical reaction cycle, and we introduced that reaction cycle idea that we copied from biology, simplified all the way down, coupled that to dynamic, chemically fueled self-assembly. I think these are the first steps towards single molecule transition. I showed the first steps towards single molecule transitions. These are very small steps getting closer to realizing such a synthetic molecular machine that does actually a very sophisticated function. If we are okay on time, then I would like to talk about the applications. If not, then please cut me off.
Let's carry on.
Let's carry on. Okay. When it comes to applications, I think as we heard before, I think it's very difficult to kind of like predict, oh, this is where it's going. At least for me, it is. Maybe not been in this field for long enough, but it's very difficult to see, I'm going to make this kind of new function. What I'm usually looking for is something that is weird and something that is unpredictable. And then a function will come out of that and an application will come out of that. So one weird function we found was the following, I think, is if we add a chemical fuel to the system, we get this emulsion, and these are oil droplets. And the oil droplets are slightly different than the classic droplets. The surprise here was if we look at the absorption and if we look at the HPLC profiles of the activated state, the linear decay of these molecules. So this is a molecule that just decays for hours and hours and hours with almost straight lines. And it's the same for the anhydride concentration. In this case, it just decays linearly. Zero order decay profile. So we started digging, where does it come from? Well, here's the concentration of anhydride. Where does it come from? We apply fuel, we get our active state. Look at the red line, active state. We run out of fuel, but the droplets survive, but they will die. But they will die with extremely linear kinetics over the course of 3 hours into this linear kinetics. And where does it come from? Turns out these droplets, they protect their components from hydrolysis. So we need water in order to hydrolyze our molecules. In such an oil droplet, there is no water because it's phase separation. There's almost no water inside. We're talking about a couple of molecules of water inside. So the hydrolysis only takes place on the fraction that remains in solution, which is equal to the solubility. So that means that if you take a molecule that is even less soluble, C10, 2 extra carbons, or an order of magnitude less soluble, the slope gets even flatter. So we can tune the slope by the solubility. And we can set this lifetime of such an emulsion by the amount of chemical fuel that we add. It gives us a lot of control over how long a material is active. And not only that, it also gives you control over how fast it decays. And then finally, most stuff decays with first order kinetics. This decays with zero order kinetics. So every hour, you get the same decay profile. So we started thinking, okay, what kind of application can we think of? The immediate thing that came to mind is drug delivery. So a material that releases or that decays with linear kinetics over the course of hours. If you now load that up with drugs, that's what we did. We add our fuel, we add our drug, we load up the droplets with drugs, we trap them in a hydrogel, all the fuel is now gone. We add a buffer and we measure the concentration of drug that has been released from these droplets. And we get this extremely linear decay profile that correlates with the extremely linear decay profile of the oil droplets. We don't do that with the oil droplets. It releases the first order kinetics within an hour. And now we're just sitting there almost 24 hours and we have this linear decay. That means that as a patient, if you apply such a hydrogel to whatever your skin or whatsoever, it releases the same amount of drugs every hour for 24 hours. So it means you don't have to take a pill every 3 hours, but it also means that you don't have risk of overdoses or underdoses because you have a first release in the beginning. You have this linear release. So it's an application that came out of chemically fueled emulsions, which I would have never thought of in the beginning. And I think for me, if you want to go for applications, I think being able to scale up is as important as being able to scale up your systems is extremely important. It allows you to actually start exploring material properties of a system. So these oils that I talked about, you can order them in liter scale. It actually turns out you don't need the chemical fuel. You can just start with the oil, sonicate it into tiny droplets, add a gel, and then you have your drug release. So this is an application of such a chemically fueled system. We published it, we patented it. And yes, we're working with anhydrides, which are not the nicest molecules. They don't get out of the gel though. So that's something to think of. Because of the hydrolysis reaction, they never make it out of the hydrogel. And we're now also looking at other molecules that are less aggressive, but still hydrolyzed with zero kinetics. Okay, with that, I would like to finish up, but before thanking my wonderful team, all the people that helped me over the past years, and of course, my funding agencies listed below. And I'd like to thank you all for your attention.
Yeah, thanks very much. It was some really fascinating stuff there. And we've had some questions come in the chat. So I don't know if you can see them, you can engage them if you want, but if you want me to read them out, I'm also happy to do that.
Let's go ahead and read them out. I think it's more dynamic.
Yeah, sure. So one was, have you used a fuel gradient to study daughter-parent dynamics?
That is a good question. Have you studied fuel gradients? Yes. Daughter-parent dynamics? Not yet. That would be really cool. That's a great idea. I do not know if the diffusion of fuel is much faster than the motion of daughter and the dynamics of this division, etc. So I don't know how long we can sustain such a gradient, but that is a really good idea. Something we should definitely. So no, I did not, but I like the idea a lot.
And then one was, do you have a feel for how much energy from your fuel is harnessed by your systems versus how much is dissipated, presumably as heat, which is in brackets?
No, also no. There's a few things we do know. The reaction cycle itself in forming the anhydrides is extremely efficient in the sense like the EDC, like ATP, does not really hydrolyze spontaneously. The EDC will hydrolyze, but about 98% of it goes through the anhydride state. So it is efficient in that sense. Yes, we lose 2% of the EDC just by direct hydrolysis, and that's just completely wasted. So that's already 2% loss. Now that question then depends on what are we doing with it. So we have molecules that are just active and then don't do anything and then open up again. So they just float around in their activated state and then die. So then in that case, the answer would be 100% is wasted. 100% is wasted in release of heat without any function. In the case of the self-assembly, that's something we did not study. It's not something we studied, but it's something we're really interested in measuring, first of all, how much heat is released upon just hydrolyzing this molecule and how much of that actually is used in folding a molecule and changing its properties. But that would, of course, depend on each state of each system individually.
Then this one's a bit of a longer one, but hopefully it'll come to you. So I know coacervate behavior can be profoundly changed by changes in solution ionic strength. Is this something that can be exploited in the context of these reaction cycles? So, for example, by using reactions that raise or lower the ionic strength of the solution.
And thereby changing the coacervate properties. it's not something we have looked at, because basically what you're doing then, which is also beautiful, is you're changing the environment, and that induces conservation, or that dissolves your droplets, as opposed to the chemical reaction is actually part of the environment. So what we were after in this specific case is molecules really flow into that droplet and then flow out of the droplet. So the droplet is controlled by kinetics of inflow and outflow, and not so much by raising a concentration of salt and then decreasing the concentration of salt. But I think that's a brilliant idea too. It's not something we have looked at so much. So I could imagine, by creating salt by a chemical reaction cycle and dissolving that salt, again, you could induce droplet formation simply by going from one state in the system to another phase. But it's not something we have looked at.
And then there were some nice compliments that sort of flowed down there after that. And then at the bottom, there was a comparison somewhere. They said the hinging of a hemoglobin protein related to pH swings affecting O2 binding release and CO2 binding release would be a good natural example. I think that was related to your final applications.
Yeah.
So I don't know if anyone else has any other questions, drop them in the chat and I'll keep engaged. Or you can hands up or just unmute yourself and ask anything. I have one, at least as I'll actually have a couple. How does your microfluidics experiment work exactly? Is it one of these things where you produce a blob and it basically goes down a channel? Is that what it is? And then you can look at the blob and that's basically how it works.
So it's a little bit more complicated because you cannot look at a blob because it moves very fast. If by blob you mean a water droplet in an oil flow. So what we do is we have two water flows that come together. One contains a precursor and the RNA and all the other ingredients, and the other one contains fuel. So they hit they come together. And then that combined flow is captured in a oil and produces droplets. So basically that is flowed into oil flow, and that creates a droplet every 100 milliseconds or something like that. And these flow so quickly that you cannot image them. So we capture each droplets in a setup that captures droplets. And that is made for that is basically a kind of container that the oil can still flow around, but it holds the droplets in place. And that allows us to immediately start imaging. So that allows us also to image as quickly as we do. So the moment that we mix stuff together, the moment that we start up the pumps, droplets are being produced. And then we just let the system equilibrate for 10 minutes. And then we grab the first droplet that is being produced. And we know the age of that droplet is less than a second or two, which is very different if you're mixing in bulk. You mix stuff together, you shake it, and you put it on the microscope. You have to find your focus. You already spent 15 seconds at minimum before you have your focus and then the first droplets have already appeared. So this was brilliant for us. Within a matter of seconds, we could just start imaging. That allows us to capture the emergence, the whole process of the system.
And then my other one was, so in the examples you showed of the droplets that grow, they just get bigger and bigger and bigger, and they consume things that are around them, and they keep sort of basically aggregating, and then eventually the largest one dies last.
Yeah.
Have you tried, or do you have a feeling for how, can you control the size of these? Can you set an upper limit to how big you want the system to be? I think, because I work with things like vesicles where you can, obviously size in different ways, but you can control curvature depending on what the lipid looks like. And I imagine your molecules can do the same thing. Can you do that? Is that something that matters? Do you care about it?
Yeah. A lot. Size controlling molecular samples by chemical reactions. We care a lot about this. So in this specific experiment, you could just add less building blocks, and thereby the growth in this particular case is mostly by fusion. So that means that in the end, you will only end up with a few droplets. And I think by adding just less stuff, you'll get less droplets, and therefore you have a final smaller droplet. But it's kind of cheating. There's just less of stuff available, and therefore it will all end up in one, and therefore you have kind of size control. Now, the reason why we're super interested in this kind of droplets is because there's a theory paper out there that says if your inflow of droplets, if your molecules are flowing in from the outside, then the inflow of material for each droplet scales with its surface area. The larger the surface, the more stuff will flow in, because statistically the chance that you get hit by a molecule is larger. If your outflow of molecules scales with the chemical reaction where you're deactivating molecules, so if the deactivation happens inside, then that scales with the volume. That's a chemical reaction, and the chemical reaction scales with the volume. The larger the volume, the faster the reaction. So your inflow scales with surface area. The outflow first order reaction scales with volume. So that means that there is an optimal radius or an optimal volume or an optimal surface area. That means that all the droplets should want to become one size. If they are too large, they shrink because they don't get enough material in and they lose a lot of material. If they're too tiny, they will grow like crazy because they don't lose enough material. And that's what we're after in this case. If we're now creating this in steady state, that means that the chemical reaction cycle, the deactivation in particular, is dominating the droplet size. The faster the deactivation, the smaller this critical radius. So that means that by wasting energy, you could control size of an emulsion, which is against the principles of fusion, which is favored, against the principles of Oswald ripening, where Oswald ripening says everything must become one large droplet. So we're super excited about this whole theory paper, and looking at these kind of principles. Can chemical reactions, the wasting of energy, can that be used for something that biology is extremely interested in, of course, like how you control the size of an object? One hand, less material available, make sure that everything ends up in one. I think it's kind of cheating. On the other hand, through chemical reactions.
So yeah, thanks. That's good. So when that's a theory paper that's out there right now, is it? Or? Yeah.
Okay. Frank Hüdiger, team in Dresden, and they're working on literally these ideas. A becomes B, B phase separates, B becomes A, but that happens in the droplet. What happens to the droplet properties? Amazingly, if you now have I think this is amazing work, and it's in theory. If you now have all of these droplets at the critical volume, they're all happy. They're all the same size, right? 100 or 1 micrometer in size. You have 100 droplets of 1 micrometer in size. Now you double the energy influx, you double the amount of material available. How do you get 200 droplets of the ultimate volume? That's where the system wants to go to. Like, they want to go to the critical volume. There's two ways. Either you nucleate new droplets, that comes with an energy penalty. Nucleation costs energy, or you start splitting off the other droplets from the other droplets. You basically grow a little bit beyond this critical volume, and then you start pinching and breaking in two. And basically, they have another theory paper, Nature physics paper, that says this is possible, provided that they're at the optimal volume, and you basically suddenly surge to the next level, and you have relatively unstable droplets or low interfacial surface area, and then this can happen. So super exciting stuff. It's just a shame that it's all in theory. We like to build a system around that, of course.
You got to start somewhere. Someone's actually asked if you could type those references into the chat, which I think some of us would appreciate if you can. There is another question that I'll ask you if you're good at multitasking while you're doing that, which is, might you be able to stabilize imaging by using a slow laminar flow, so oil on top of aqueous?
Yeah, sorry, I'm not good at multitasking.
Okay, that's fine. We can.
I did type the name, and then I heard laminar flow. Sorry.
Yeah, so it was might you be able to stabilize imaging by using a slow laminar flow? So, for example, oil on top of an aqueous solution.
Stabilize the imaging part in the microfluidics.
I presume that's what the question pertains to. Yes, but
if that's not, just speak up in the microphone. Yeah, I think so. But in order to make the microfluidic droplets, you need pretty fast flows. Oh, okay. You could have the flows like Ayusman was showing earlier, having the flows next to each other and then see the droplets emerge through such a gradient. I think that's actually a good idea. I'm always scared that when you do self-assembly in such microfluidic devices, I'm scared of clogging the devices, and then that makes it complicated. It's not something we have tried. So having like fuel come in and then precursor come in and just let that flow next to each other and see getting droplets go from one side to the other. That would be pretty cool to study as well. Yeah.
I was thinking of possibly using ultrasound as a way of creating the intermixing of your reagents, where you could have it flowing horizontally through a tube where the interface would be stable, and you would be able to move your camera along the tube to, in a sense, create a time, image.
Yeah, I guess that would be possible. Like, you need a high-speed camera and a fast-moving camera, of course. Yeah, I guess that's possible. I'm assuming this has been done as well, but I'm definitely not my expertise. But these are interesting ideas.
So one thing I suddenly thought of, but I'll have to check something. So you showed your materials horizons. That was obviously one of your papers with like the drug delivery. What conditions is this? What scenario is the drug delivery experiment performed in exactly there? So if I'll add some context, which is my original thought, and I realized that you might have already answered by the example you showed, is you obviously look at all this chemistry inside a droplet. And I wonder what the dynamics in the droplet are like versus in, say, a larger system. Or is the droplet in your microfluidics experiment significant enough in size that the general things of like convection I'm not saying you can account for turbulence, which is generally regarded as being something that you can't really predict, but does it map well to a much bigger system when you might use it in drug delivery, or is it still idealized?
It's exactly the reason why we lock up these droplets in a hydrogel, because the moment that you don't do that, you will have Oswald ripening, first of all. So the droplets get larger and larger. And then you have either creaming, which means the droplets start floating, or they have sedimentation, or the droplets start sinking depending on the density. So you have all of these kind of problems, and this will happen over the course of hours. So in our experiments, this will definitely kill the whole idea. So we're trapping these droplets in the hydrogel, just a very simple hydrogel, to avoid all of these complications. And while I think you would still have some Oswald ripening, it's severely hindered. And then the emulsion just becomes more of a material. And we have not studied the droplet dynamics within such emulsions with much detail. Within such hydrogels in much detail, but we have without the hydrogel. And without the hydrogel, we have a lot of, as I said, Oswald ripening. In fact, Oswald ripening is accelerated by our chemical reaction cycle. So this whole idea of small droplets are shrinking and big droplets are growing, because that's theoretically more favored, is actually accelerated by the chemistry of our chemical reaction cycle. We see turbulence, but you see turbulence everywhere where you start studying things in bulk. So this is very difficult to say like what is real and what is not. It's an extremely complicated project. It's suspecting that droplets start sinking and that starts pushing up. So we get this kind of microscopic flow through our systems, beautiful patterns and that kind of stuff. But I find it so difficult to see what is real and what is not. So that's really where we don't have the appropriate training in this kind of field. So we have all of these other convection and turbulence and these kind of things happening. And it is exactly the reason why in order for material science, you don't want to deal with that. So if you just lock it all up in a hydrogel, it prevents also from flowing, makes it also much more applicable than working with just a solution of droplets.
Yeah, I have to apologize. I now remember that you shared that on the slide, and apparently I'd already completely forgotten it and then just asked the question that you'd already answered, but never mind.